In the next section, we will examine the role of the immune system in rheumatoid arthritis. As noted, the triggering effect that unleashes the destruction of the synovium in RA remains a mystery. Nevertheless, the factors that contribute to the dysregulation of the various arms of the immune system and the breakdown of self-tolerance are becoming clearer. Proof of the role of tissue necrosis factor alpha, or TNF-alpha, interleukin-1, and autoantibodies is provided by the use of biologic agents, which act directly to nullify the effects of these cytokines. They bring about dramatic improvement in the clinical state of patients with RA when these agents are employed early in the course of the disease. An interplay between the innate and adaptive immune systems is also important. The innate immune system recognizes structural patterns on microorganisms such as lipopolysaccharide or LPS and N-formylmethionine. These pathogen-associated molecular patterns or PAMPs are recognized by pattern recognition receptors or PRRs, carried on natural killer cells or NK cells, monocytes and macrophages, dendritic cells and neutrophils. These PRRs are encoded by the DNA and are limited in number. In contrast to the enormous repertoire of antibodies and T-cell receptors generated by the adaptive or acquired immune system, the cells of the innate immune system recognize microbial virulence or danger signals that are shared by many pathogens. When cells equipped with PRRs, macrophages, and dendritic cells recognize these genetic signals and trigger activation of the complement system and production of various inflammatory cytokines. While this recognition system, in conjunction with the neutrophil killing, are quite effective, the recognition system lacks specificity for unusual organisms that can only be sought out by the acquired immune system. Nevertheless, cancer, chemotherapeutic agents can cause dangerous, even life-threatening neutropenia, testifying to the vital role played by elements of the innate immune system in protecting against virulent microbes. As important as the innate system may be as a baseline defense mechanism, it is equally, if not more important, in its interaction with the adaptive immune system, which provides a way to arouse the more selective and powerful adaptive system. Paramount among these interactions are the toll-like receptor TLR proteins, which are the LPS CD14 receptor, which signals dendritic cells, macrophages, and epithelial cells to produce inflammatory cytokines and upregulates effector molecules on the cells bearing the LPS receptor, and to activate T and B cells to initiate the antigen-specific adaptive immune response. Remember, LPS is a component of the cell wall of gram-negative bacteria and is often called endotoxin. It is important to recognize that when massive amounts of LPS are recognized by TLR4, this leads to release of a shower of cytokines that can precipitate shock. Cytokines are small soluble proteins produced by hematopoietic and non-hematopoietic cells. Cytokines are the effector molecules for the innate and adaptive immune responses, the production and expression of which can go awry in many inflammatory and autoimmune states. There are at least 20 cytokines, and as a class, the cytokines are involved in the regulation of the growth, development, and activation of T and B cells, and in mounting the inflammatory response. Cytokines that primarily target leukocytes are called interleukins. Other cytokines are colony-stimulating factors. The cytokines include TNF-alpha, which plays a central role in the pathogenesis of RA. TNF-alpha causes fever, capillary leakage, enhances NK cell function, increases production of acute phase reactants and induces production of a number of pro-inflammatory cytokines. Thus, the multifarious functions of tissue necrosis factor alpha, if not controlled, could cause significant harm to the individual. It should be made clear that cytokines activate gene transcription by promoting growth, differentiation, and activation of effector cells of the innate and acquired immune systems. Numerous studies have shown that cytokines use the JAK-STAT signaling system, JAK-STAT signaling system, to upregulate gene expression. Growth hormone and prolactin also use the system of intracellular signaling. JAKs are a family of tyrosine kinases which, after undergoing autophosphorylation, activate the signal transducers and activators of transcription. So this is STAT, S-T-A-T, signal transducers and activators of transcription, family of transcription factors. Thus, as opposed to receptor tyrosine kinases, the JAK system, JAK system, employs separate tyrosine kinases and transcription factors, STATs, to effect transcription. The adaptive, acquired, or immune system, let's talk about that. In fact, let's talk about that in the next video.